So when an individual inherits a heterozygous type of sickle cell, here mostly they are just a carrier. Okay, they don't show symptoms of sickle cell, only under extreme physical conditions. But then there's what we call a case of selective advantage, where these human beings actually are resistant to malaria that is caused by the Plasmodium falciparum. Okay, so there are more than 900 mutations in the genes that code for the alpha and beta chains. Okay, these are proteins. Majority lead to a few pathological phenotypes. For example, of thalassemia, this is just an absence of either partial or the total absence of one or more of the alpha or beta chains of the hemoglobin. Okay, so certain mutant hemoglobins are common in many populations. The patient may inherit more than one type. Then you also have what we call glycated hemoglobin. We usually measure this one. We can measure this one, glycated hemoglobin. It provides a valuable information for the management of diabetes mellitus, because here we take advantage of the 120 days lifespan of the red blood cell, which remains once it is glycosylated with glucose. Okay, so the glucose can stay with the red blood cell, okay, for 120 days. So glucose enters these red blood cells and it is glycosylated with some amino acids such as lysine. Okay, so the fraction of the glycosylated hemoglobin, normally about 5%, is proportional to the blood glucose. So you can use it to actually it gives an information how the person is managing their diabetes. Okay, also you also have now plasma proteins. These are proteins that are found in plasma. Okay, so you have albumin, you also have immunoglobulins, and also fibrinogen. Okay, these are for immunity. Fibrinogen used for clotting factors. Okay, the process of clotting. Albumin, it acts as a transit. It helps in transporting things that are fatty in nature, fatty acids, steroid hormones. Okay, anything that is lipid in nature, for it to be transported, it has to bind to this protein called albumin. So most of all glycoproteins, okay, occur widely. Bacteria, even humans, perform numerous functions. Okay, so proteins of cellular membranes, blood group substance, hormones. Okay, also these hormones such as the gonadotrophins are glycoproteins, and that's the functions of certain types of glycoproteins. Okay, collagen does a structural function. You have transferrin and ceruloplasmin for transport. Alkaline phosphatase, it's an enzyme, hormone, and drug action. Most receptors, and also hormones such as thyroid stimulating hormone. So the linkage here is called the O-linked. It occurs between proteins and glucose. For example, this one right here is a glucose molecule. It attaches itself to a protein. This is another glucose molecule attached to a protein. These are conjugated type of proteins that we talked about. So plasma proteins are found in plasma serum. Okay, they are classified, okay, based on how they are synthesized. So most of the plasma proteins are synthesized in the in the liver. Okay, for example, albumin, most are synthesized in the liver, synthesized by plasma cells of the bone marrow. Such, okay, so others like immunoglobulin are synthesized in the bone marrow. Okay, so number of plasma proteins that bind to certain ligands with high affinity and specificity. So for these proteins, the way they bind to certain ligand it needs to have high affinity for that ligand, and it must be specific to that certain type of ligand. So as a result of this, therefore, they act as a reservoir for ligand control. Okay. In albumin, it's predominantly a plasma protein. It's not an enzyme, no, a hormone, and its total maybe the range that we expect to find in the body is 35 to 45 grams per liter. So it transports hydrophobic fatty acids, okay, bilirubin, and also certain drugs are transported. Availability of sites capable of being very toxic. So drugs such as the penicillin, aspirin, and also the sulfonamide drugs are also bind to albumin. So the clinical importance is that when there's a decrease in albumin, okay, it shows that there is malnutrition. Also, you have liver cirrhosis, which has to do with the albumin synthesis, and also the nephrotic syndrome, where this is just a syndrome, has to do with the kidneys malfunctioning, where albumin is excessively lost in the urine. Metal ions, major importance of proteins in binding and transporting metal ions. For example, metal ions such as iron that are bounded to certain proteins such as the myoglobin. So certain proteins can just they bind to certain metal ions, and one that is common is iron, which is found in hemoglobin and myoglobin, also in certain proteins called cytochromes. So within the plasma, iron is transported by another protein called transferrin. So it will deposit iron deposited in storage sites. So transferrin is a transporter, but then a storage form is ferritin. Okay. So concentration of ferritin in plasma is proportional to the amount of stored iron. Okay. So the binding protects against toxic effects. So it has to bind to. Then you have ceruloplasmin, a major transport protein for copper. So just like you can talk of, we talked about transferrin, which transports iron. We also have ceruloplasmin, which transports copper from the liver. Okay. Increased concentration of protein may occur in active liver disease, okay, and tissue damage. So the deficiency causes a condition Wilson disease, okay. The main problem is the mutation of this gene here. Then immunoglobulins, proteins produced in response to foreign substances, and they are produced by B lymphocytes. During an acute phase reaction, there's marked increase in synthesis of these proteins. Okay, for example, the C-reactive proteins, a major component of the acute phase response, okay, synthesized in the liver. Okay, so they can also show when there is an increase in these acute phase proteins, it will show us that there is maybe an inflammation taking place in the body. So mediation of binding of foreign polysaccharides, phospholipid complex, polyanions, all these can be used in diagnosis and monitoring of infection. So now the important part that we need to know, that you need to know here, interactions. You need to understand these interactions. Covalent and non-covalent bonds. So non-covalent bonds, they contribute to the structure, stability, and functional competence of biomolecules. Interactions both within and with water. Hydrophobic interactions, these non-polar compounds. Okay, so compounds that are non-polar, they have these hydrophobic interactions. You also have electrostatic interactions. We've talked about the salt bridges. Okay, they also have the van der Waals forces. You can find them these in the tertiary structure. They are weaker as compared to hydrogen bonds. Okay, and these are just transient dipoles generated by rapid movement of electrons. Okay, so here we just need to know what's important is the functions, the clinical importance, okay, and also these interactions at each stage, okay, at the each stage of the structure, either primary, secondary, tertiary, and quaternary.